Today we are going to have a quick look at ErePOC, a method to predict protein binding pockets that are more suitable for DNA-encoded library screening. DNA-encoded libraries can contain huge numbers of compounds, but not every protein is equally compatible with this approach. This study asks a simple but important question. What kind of binding pockets are most likely to work well with such screening strategy? DNA-encoded libraries, or DEL, represent a powerful screening technology for finding small molecules that bind to protein targets. The approach can test enormous chemical libraries, often containing billions to trillions of compounds, in a highly efficient way. But despite this huge screening power, only a limited number of DEL-derived molecules have progressed to clinical trials or reached the market. Why is that? Part of the reason is that DEL is still a relatively young technology in mainstream drug discovery. But there are also deeper challenges. DEL data can be difficult to interpret, DEL hits are not always easy to optimize, and we still do not fully understand what makes a protein target truly suitable for DEL screening. To address this, researchers have started combining DEL approaches with artificial intelligence. In this study, the authors focus on one key question: what are the characteristics of the protein binding pockets that successfully interact with DEL molecules? By answering that question, they hope to improve target selection and make DEL-based drug discovery more efficient and more successful. When the authors compared different classes of binding pockets, DEL pockets stood out. They were generally larger and showed stronger hydrophobic character than typical ligand-binding pockets. So even before building the model, the data was already suggesting that DEL molecules may favor a specific pocket environment, one that is bigger, more exposed, and more hydrophobic. The authors then developed a model designed to learn what makes one binding pocket functionally similar to another. Starting from structure-defined pocket residues, they encoded each pocket using sequence-based ESM2 embeddings and then applied contrastive learning so that pockets binding chemically similar ligands would be placed closer together in a learned latent space. That is really the key idea. The method is not simply comparing pocket shape, but instead learning a more function-aware representation of the pocket. When the authors tested the model, it separated different pocket classes much more clearly than ESM2 alone, while also generalizing well to unseen examples. This is what allowed the model to become the core engine for searching DEL-compatible pockets across much larger protein space. When the authors visualized the pocket representations, the model separated different pocket classes much more clearly than ESM2 alone. To sum up, DEL-compatible pockets seem to carry a learnable signature, and the model uses that signal to predict promising targets for DEL screening. By combining AlphaFold predictive structures, pocket detection, and their learned pocket representation model, the authors identified more than 2,700 DEL-compatible proteins across the human proteome. That is it for today. I hope this tool can be useful for your research and for exploring new opportunities in DEL screening. Until next time, take care.